Hello there. In this video presentation, we will be focusing our attention on a technique used in the laboratory setting. This technique is known as an enzyme-linked immunosorbent assay, or ELISA for short. Although this method is mainly known as ELISA, it can also be found under the name of enzymatic immunoassay, or EIA. As we go through this presentation, we will be focusing on a variety of key topics which include the discussion of the general principles involved in ELISA, the identification and discussion of the different varieties of ELISA techniques, and an example of application of the ELISA technique in scientific research will be presented. So what is an ELISA and what specifically is it used for? An enzyme-linked immunosorbent assay is a test that is able to detect and measure antibodies that are present within an individual's bloodstream. It is important to note that antibodies are proteins that the body produces in response to substances called antigens. These antibodies present in the bloodstream can be related to certain infectious conditions which can be diagnosed through ELISA testing. Some conditions include, but are not limited to, HIV, Lyme disease, pernicious anemia, rotavirus, and syphilis, just to name a few. Let's jump a little deeper into the general principles of ELISA. ELISA combines the specificity of antibodies with the sensitivity of simple enzyme assays. This is accomplished by using antibodies or antigens that are coupled to an easily assayed enzyme. The ELISA can then provide measurements of both antibody and antigen concentrations. It is important to note that ELISA can be used through two main variations. One, to detect the presence of antigens that are recognized by an antibody, or it can be used to test for antibodies that recognize an antigen. We move our attention over to the different types of ELISA. There are 4 different types of ELISA, and these types include indirect ELISA, sandwich ELISA, direct ELISA, and competitive ELISA. Let's now take a look at the differences between each ELISA test. In indirect ELISA, both a primary antibody and a secondary antibody is used, but in this case, the secondary antibody is not labeled with an enzyme. The secondary antibody is the one labeled with an enzyme in this case. In a sandwich ELISA, the antibody is immobilized to the plate, and this antibody is then known as a capture antibody. In addition to this antibody, there is also the use of detection antibodies, which usually include the unlabeled primary detection antibody. In direct ELISA, only an enzyme-labeled primary antibody is used. This essentially means that no secondary antibodies are needed for the rest of this test. As opposed to the 3 ELISAs mentioned before, competitive ELISA is much more complex because it involves the use of an inhibitor antigen. In competitive ELISA, the inhibitor antigen and the antigen of interest compete for binding to the primary antibody. Although there are 4 different types of ELISA testing, most of them fall under these 5 general procedural steps. The steps go as follows. First, the wells in a microtiter plate are coated with antigens. Then, all unbound sites are blocked to prevent a false positive result. A primary antibody is then added to the wells, followed by an addition of a secondary antibody that is conjugated to an enzyme. In the final step, there will be a reaction of a substrate with the enzyme to produce a colored product. This product is what indicates a positive reaction in an ELISA test. We will now be moving over to an example of application of technique of ELISA which was found in a scientific article.
Hello, welcome to the last part of the ELISA instructional video. We will be analyzing the application of ELISA in HIV testing. ELISA stands for Enzyme-linked immunosorbent assay. As you have probably seen earlier in the video, ELISA consists of mainly two core functions. These core functions are as following: the use of antibodies to bind a specific target antigen of interest, and the use of an enzyme to detect the presence of that antibody. Here's some quick facts and methods. In addition, this method is rather clear and simple. The ELISA method is the most commonly used for HIV testing. It starts off by gathering a sample of the patient's blood for the finding of antibodies. This blood sample will be added to a cassette that contains a viral protein, also known as antigen. This testing will be checking for the response of a certain protein that body makes in response to HIV. If tests come back positive, it has a 99.9% accuracy. The enzyme substrate complex is a temporary molecule formed when an enzyme comes in perfect contact with its substrate. The substrate causes a conformational change or shape change when the substrate enters the active site. Now, antigens are substrates that cause the body to produce antibodies, such as the viral protein. Antibodies bind antigens very specifically like a lock and key, neutralizing the HIV virus and preventing its further spread. Plate is a flat plate with multiple wells used as small test tubes. The following picture is an actual microtator used in lab, and the following image is how I will be representing it on the board. Here's a quick breakdown of the complex. If you would zoom in on one of the microtiter plates, it will be containing the following. The wells are covered with specific type of antibodies for HIV. The antibodies would be GP41 and GP120. The antibodies are represented by the blue white-looking structures. These are free to react. Following, we'll be adding the antigens. These antigens are represented by the orange triangles. These antigens will interact with the antibodies at the bottom of the wells. These can be seen with the following structure where the antibodies are not attached to the orange antigens. The HIV antigen is also known as P24. Another part of this reaction is to add another type of antibody, which is represented by the pink figures, but this time it contains an enzyme-linked, which in the circle figure on the pink enzyme. These enzymes can interact with a substrate to convert color and be visible. The antibody along with the enzyme link will attach to the antigen, which is represented by the collaboration of the pink and blue figures. Now we will be adding substrate, which is expressed in the circular yellow figures, and will allow some discoloration in the wells to visualize. Now this is a general idea to find out if the antibody is present in the serum. If the antigens react, the person will be positive for HIV. Thank you for watching and stay safe.